how to stop neurodegeneration from occurring. Hello, my name is Shai Berlin. I'm an assistant professor at the Faculty of Medicine here at the Technion. I'm part of the neuroscience department, and my lab focuses on NMDA receptors, both in health and disease. We're both interested in understanding how the receptors function in cells, what different subunits and different channel compositions do, and at the same time we're also exploring different mutations and how these instigate different diseases of the brain. We're using various tools and methods, especially genetic tools, and we are very hopeful to be able to develop new small molecules or small peptides to be able to stop neurons from dying, as well as we're using these very new state-of-the-art genetic techniques where we can kind of assemble subunits at our will, meaning we can form different type of receptors, and then we're able to say exactly how these different channel compositions behave in cells. One tool is to be able to tag different receptor subtypes, precise NMDA receptor subtypes with these light-sensitive molecules, and then we can switch them on and off at will, and then we can say precisely which subunit or which channel subtype is doing its activity at the synapse. So toxins have been naturally evolved over millions of years, and these small proteins naturally attack different NMDA receptor subtypes, and so we have a perfect tool to target different subtypes of NMDA receptors. So when we combine these naturally occurring toxins with fluorescence moieties or different chemical conjugated moieties, we can really kind of bring together two different fields, so we can look at the toxins, target different subunits in real cells using live microscopy combined with electrophysiology, and so this is really bringing together two different fields of study. Whereas antibodies are really good, high affinity, and we can play around, and there's a variety that are sold both by Alamon and other companies. We're still looking for smaller reagents. Toxins are kind of the natural next step for us because of their smaller size, very high affinity. Some toxins even penetrate the cells, and so these can actually provide us like an added benefit over antibodies. I see a great potential for combining both labeled antibodies and labeled toxins in live cell imaging for different assays. I can see different antibodies targeting different ion channels and different subunits, whereas toxins labeled or conjugated to different chemical moieties can be targeted for additional targets. There's a huge potential, especially for targets that don't have antibodies or for which we have not produced high affinity antibodies, and nature has done this for us by producing very high affinity toxins for various, if not all, different channels, especially NMDA receptors. I think anyone who's interested in physiology, ion channel physiology, electrophysiology, live cell imaging, I think those are the labs that really should be using conjugated, labeled or non-labeled fluorescent toxins. These can be used to localize your target, to see where your ion channels and targets are localized on top of the cells. Of course, they can be applied to control the activity of ion channels. So I really think these are incredibly powerful tools for anyone who's interested in ion channels. First and foremost, because of performance of the reagents, we obtain consistent results over time, regardless the lot, regardless the different batches that we purchase. Great tech support, or any support in general. Everything is sent very quickly to us via FedEx or any other types of courier. But I would say mostly because of the performance. We're very, very impressed with the reagents' performance. I would like to have a palette of antibodies targeting only the extracellular parts for all the NMDA receptor subunits. Same goes for toxins. I would like to have specific high affinity toxins for different NMDA receptor subtypes. If there are additional tools that we don't know of and you're willing to produce for us, we will take them all. We're looking for talented PhDs and postdocs to join our group using methods such as electrophysiology, live confocal microscopy, and molecular biology. And we would be happy to collaborate with any lab that is also interested in similar aims as we do, ion channels, physiology, in particular NMDA receptors. So please contact us.